Welcome to our webinar. My name is Vanessa Rogoshak, and along with my friends and fellow medical genomics students at the University of Toronto, Nihal Al-Menabawi and Aisha Kidwai, we have put together a presentation explaining the process of genetic testing for neurofibromatosis conditions. Let's start with a brief overlook of what we're going to be discussing today, beginning with signs and symptoms of neurofibromatosis, inheritance patterns, diagnostic criteria and alternative diagnoses, before moving on to genetic testing, its benefits and possible results, and finishing off with treatment and screening options available to patients with neurofibromatosis. So far, I've been saying neurofibromatosis, but this is actually an umbrella term that encompasses a set of 3 distinct genetic disorders, including neurofibromatosis type 1, neurofibromatosis type 2, and Schwannomatosis. All of these disorders can cause benign and cancerous tumors in the brain, spinal cord, and the nerves, and can affect multiple organ systems, including the musculoskeletal system, the endocrine system, and the skin. For the first part of the presentation, we will be focusing on neurofibromatosis type 1, or NF1 for short. This is because this is the most common neurofibromatosis condition. How common is NF1 exactly? It affects about 1 in 3,000 individuals, and while this might not seem like a lot at all, NF1 is actually considered one of the most common genetic disorders. Let's briefly discuss what I mean when I say NF1 is a genetic disorder. NF1 is caused by a mutation or a genetic change in the NF1 gene. This gene normally functions to protect the nervous system against tumors. When people have a mutated copy of the NF1 gene, it leads to the symptoms of the condition. But how can someone acquire these mutations? There are 2 ways someone can acquire a NF1 mutation. The majority of NF1 patients acquire a mutated copy of the gene that causes the disease from an affected parent. This means that the condition is inherited. So an affected parent passes on the mutation to a child. However, between 30 and 50% of cases are de novo, meaning that they are caused by spontaneous mutations in the NF1 gene that occurred in the sperm or egg cell that became the individual. This means that the mutation is not present in either parent. It is only present in the affected person. However, that individual can still pass on the mutation to their own children. NF1 is inherited in an autosomal dominant manner, meaning that each child of an affected parent has a 50% chance of inheriting the mutation and developing the disease. Let's move on to some of the symptoms of NF1. NF1 can be tricky to treat and diagnose because signs and symptoms can be variable, progressive, and unpredictable. In addition, symptoms can range from very mild with barely any signs, to moderate, to more severe with complications. Often, people with mild or moderate forms of NF1 don't know they have the condition and may not seek medical help for many years. Keep this in mind if you're wondering if you or your family are affected by NF1. Some common symptoms of NF1 include the presence of flat, light brown spots on the skin, known as café au lait spots. Having more than 6 of these spots is suggestive of NF1. Another symptom can be tiny bumps on the iris of the eye, known as Legius nodules. Some more severe symptoms include benign tumors under the skin. These are known as neurofibromas and are what the condition is named after, as well as bone deformities, freckling in the armpits and groin, tumors known as gliomas on the optic nerve that can sometimes lead to blindness, and in some cases, learning disabilities. In addition, arguably the most severe potential symptom of NF1 is cancer, though this is rare. It is estimated that 3 to 5% of people with NF1 will develop cancerous tumors in their lifetime. These usually arise from the neurofibromas under the skin and are due to the role of the NF1 gene in preventing cancer. And it means that NF1 patients are at a higher risk of developing a range of cancers, including brain tumors, leukemia, colon cancer, breast cancer, and some types of soft tissue cancers. We'll come back to how to screen for these cancers when we discuss treatment and management strategies towards the end of the webinar, so sit tight. You might be asking yourself, some of these signs and symptoms apply to me. Do I have NF1? Should I be tested for it? Let's explore some of these questions together. To be clinically diagnosed with NF1, a patient needs to meet certain diagnostic criteria. 2 of the following symptoms must be present. 6 or more cafe au lait spots over 5 millimeters before puberty or 15 millimeters after puberty, freckling in the armpit or groin, 2 or more neurofibromas, which you'll remember are the tumors under the skin, an optic glioma, meaning a tumor in the eye, 2 or more Lich nodules, or certain bone malformations, such as abnormalities in the skull or thinning of the long bones. Let's transition into a discussion about genetic testing and some reasons why genetic testing might be recommended for someone with or suspected to have NF1. Firstly, for a child of a parent who meets diagnostic criteria, a geneticist or genetic counselor may recommend they receive genetic testing to obtain an early diagnosis, whether or not they present with symptoms. Secondly, genetic testing could help inform family members of affected individuals so that they can also have access to testing and medical attention. Some patients who might be recommended for genetic testing are individuals who are suspected to have neurofibromatosis type 1, but a clinical diagnosis is difficult to make, perhaps because of mild symptoms or other indications. Your doctor might have sent you our way and referred you to genetic testing even if you already have a clinical diagnosis for NF1, because of all the benefits of genetic testing we've discussed so far. Should you still go for genetic testing? Receiving a genetic diagnosis, meaning confirming there's a disease-causing mutation in the NF1 gene, may help guide clinical management and can help inform other family members of a genetic disorder in the family. Please know that genetic testing is never mandatory, and a genetic counselor or genetic clinician can help you decide if testing is right for you or your family. Okay, if you're potentially interested in genetic testing for NF1, you may have some questions about what a genetic test is looking for. Let's learn more about this together. As mentioned before, a genetic test is looking for a mutation in the NF1 gene. A genetic counselor or clinician will send you for a blood draw, and your DNA will subsequently be extracted from your blood cells and analyzed. An individual has 2 copies of the NF1 gene, one inherited from their mother and the other from their father. Both copies of the gene are checked for possible changes. A genetic specialist compares the 2 copies of the individual's NF1 gene to a normal NF1 gene. If there are differences, like in this picture on the right, the specialist decides if these changes are disease-causing and point to an NF1 diagnosis. On the left, there are no mutations, and this copy of NF1 is normal. On the right, a mutation has been identified as disease-causing. Given this individual has 1 mutated copy of NF1, they are genetically diagnosed with NF1. Once a specific disease-causing mutation has been identified, any family members who wish to undergo genetic testing will only be tested to see if they have that specific familial mutation. We think it's important to go over some more information about the NF1 gene to better understand why it has such an impact on human health. As briefly mentioned before, the NF1 gene codes for a protein called neurofibromin. This means that the instructions that tell your body how to make neurofibromin are written in this NF1 gene. This protein normally prevents cells from growing and dividing in an uncontrolled way, and is needed for cells to remain healthy. We all have 2 copies of the NF1 gene, one inherited from our mother and one inherited from our father. If one of these copies has a mutation that stops the gene from working, the other copy serves as a backup, and our cells still divide normally. However, if another mutation spontaneously occurs in the only working copy of the gene, there are no more instructions for the neurofibromin protein, and all protein function is lost. This causes the cells to divide uncontrollably, and is what causes the characteristic neurofibroma tumors. People with NF1 are born with 1 mutated copy of the NF1 gene in each of their cells. Therefore, the probability of the healthy copy being mutated and neurofibromin being completely lost is much greater than someone with 2 working copies. Neurofibromin is produced in many types of cells, but is mostly found in nerve cells and specialized cells called oligodendrocytes and Schwann cells that surround nerves. This is why the main organ system affected by NF1 is the nervous system. More than 1,000 mutations in the NF1 gene that cause neurofibromatosis type 1 have been identified, many of which cause an extremely short version of the protein that prevent it from working properly. Jumping back to the genetic testing process, there are 3 possible results that you can receive. The test can either come back positive, negative, or uncertain. Let's discuss what each of these options mean. A positive result means a significant disease-causing mutation, known as a pathogenic mutation, has been identified in a copy of the NF1 gene. This result is considered to be consistent with a genetic diagnosis of the condition and is believed to increase the risk of developing NF1 symptoms in those who carry the mutation. A negative result means that both copies of the NF1 gene are normal, meaning that no disease-causing mutations were found, and the individual would not receive a genetic diagnosis of NF1. An uncertain result indicates that a variant of uncertain significance, a VUS, was identified. This means that a copy of the NF1 gene is not normal, and a mutation was found, but it's unclear whether this mutation increases the risk of NF1 symptoms or not, and if so, by how much. We all carry minor genetic changes in our genome, therefore the majority of genetic variants are not disease-causing. However, it's possible that a VUS may be reclassified and either upgraded to pathogenic or downgraded to benign, which means that the mutation does not affect the neurofibromin protein. Typically, for patients identified to carry a NF1 VUS, their medical management is based on their personal and family history of disease, and they are encouraged to reach out to their clinical team to check if there are any changes in how the mutation is classified every few years. Sometimes, someone who has a clinical diagnosis or is otherwise believed to have NF1 gets back a negative result. If your genetic counselor or geneticist believe a mutation is present, they may take additional steps to find it. The first possibility is a condition called Mosaicism. Someone who is mosaic has 2 populations of cells in their body. One group of cells has a disease-causing NF1 mutation and is affected with NF1. In this diagram, the population of cells that have the NF1 mutation are represented in blue. The other group of cells have 2 normal working copies of the NF1 gene. If your blood cells are unaffected but other cells in your body are, you may present with NF1 symptoms, especially in areas of the body that contain the cells with NF1 mutation, but still receive a negative genetic test. In these cases, it's impossible to know the extent of affected cells or where they are located. This makes it difficult to predict a person's exact risk of developing NF1 tumors or passing on the NF1 mutation to future children. In this man, his sperm cells are within the population of cells containing the NF1 mutation. He is therefore capable of passing on the NF1 mutation to his children. In this woman, her egg cells are within the population of cells that do not have the mutation, and therefore she's not able to pass the mutation on. However, since we have no way of knowing which cell populations are affected, there's no way of predicting the risk of inheriting NF1 from an affected mosaic parent. We usually say that the risk is up to 50%, but usually it's much less than that. To test for mosaicism, a genetic counselor or geneticist may recommend any tumors that were removed to be tested for NF1 mutations, and if multiple tumors are found to have an identical NF1 mutation, mosaicism can be assumed and children can be tested. Another possibility for a negative NF1 test with the presence of clinical symptoms is the genetic condition was wrongly identified to be NF1. There are similar conditions that have overlapping symptoms with NF1, and your genetic counselor or geneticist may recommend follow-up testing to eliminate the possibility of a differential diagnosis. One of these similar conditions is called Legius syndrome, a disorder characterized by multiple cafe au lait spots, learning disabilities, and freckling in the armpits and groin. Sounds familiar, right? Some additional features that may help differentiate Legius syndrome from NF1 are macrocephaly, meaning an enlarged head, and the absence of any neurofibromas or other tumor manifestations. Just like we did with NF1, let's quickly look at the gene that causes Legius syndrome, SPRED1. This gene provides instructions for making the SPRED1 protein, which is involved in a cellular pathway that controls the growth and division of cells, cell maturation, cell movement, and programmed cell death. Similar to NF1, mutations in the SPRED1 gene cause cells to divide out of control, though it is not well understood how mutations in the gene cause the signs and symptoms of Legius syndrome. This condition is also inherited in an autosomal dominant manner, meaning that an affected person has a 50% chance of passing on the mutation to each child, and one mutated copy of the SPRED1 gene is enough for a genetic diagnosis of Legius syndrome. Your geneticist or genetic counselor may also consider ordering a genetic test for the other neurofibromatosis conditions. As mentioned at the beginning of the presentation, neurofibromatosis encompasses a group of genetic disorders including NF1 and NF2 and Schwannomatosis. All of these conditions share certain characteristics, and the genetic counselor or geneticist may order a panel genetic test, meaning testing multiple genes at once, to see if the patient should be diagnosed with NF2 or Schwannomatosis instead of NF1. Let's discuss the possibility of being tested for neurofibromatosis type 2. Like NF1, NF2 is a disorder characterized by the growth of non-cancerous tumors in the nervous system. However, unlike NF1 where tumors appeared under the skin, NF2 tumors are called vestibular schwannomas or acoustic neuromas and grow in the inner ear, specifically along the nerve that carries information from the ear to the brain, the auditory nerve. These tumors can also occur in other cranial, spinal, and peripheral nerves, including in the optic nerve and within the spine. The majority of NF2 patients present with hearing loss, often accompanied by dizziness or imbalance due to the presence of the tumors within the inner ear. Other symptoms that occur due to the development of tumors include headaches and seizures, and these are because of the presence of a tumor called a meningioma that arises in the membrane surrounding the brain. Other symptoms include pain, muscle weakness, and pins and needles, especially if there are spinal tumors present. Patients may also suffer from vision problems, including cataracts or vision loss due to the tumors that affect the optic nerve. NF2 patients present with multiple skin tumors approximately 10% of the time, which leads to the possibility of a misdiagnosis of NF1. Just like with NF1 and with SPRED1, let's look at the NF2 gene and how it relates to the condition. NF2 is caused by mutations in the NF2 gene, which provides instructions for a protein called myelin. This protein is produced in the nervous system, particularly in specialized cells known as Schwann cells that wrap around and insulate nerves. Myelin is involved in several cell signaling pathways involved in controlling cell shape, growth, and the attachment of cells to one another. Just like with NF1, the NF2 gene plays a role in preventing cells from growing and dividing too fast or uncontrollably. Therefore, if both copies of the gene are lost due to a mutation, remember, one mutated copy is inherited or de novo and the other mutation occurs spontaneously, tumors can arise. Also, just like before, one mutated copy of the NF2 gene is consistent with a genetic diagnosis of NF2. And like the other conditions we've discussed so far, NF2 is inherited in an autosomal dominant manner, meaning that an affected parent has a 50% chance of passing on the mutated copy to a child. If you're worried that the symptoms of NF2 are more severe than the ones associated with NF1 and Legius syndrome, please know that NF2 is much more rare, with an estimated incidence of 1 in 33,000 people worldwide, compared to 1 in 3,000 for NF1. The final neurofibromatosis condition your genetic counselor or geneticist could recommend testing for is Schwannomatosis. This disorder is characterized by multiple non-cancerous tumors called schwannomas, which, like with NF2, develop in Schwann cells, which are cells that wrap around and insulate nerve cells. Common symptom of schwannomatosis is chronic pain, which can affect any part of the body and can range from mild to severe and be difficult to manage. Other symptoms include numbness and tingling, weakness, and headaches, depending on where the schwannomas occur. The main feature that distinguishes schwannomatosis from NF2 is the absence of inner ear tumors and the absence of any other types of tumors associated with NF2, like meningiomas. It's time to discuss the genes that cause the condition. In the case of schwannomatosis, there are two, SMARCB1 and LZTR1. The SMARCB1 gene provides the instructions for making a protein that is part of a larger protein complex called the SWI-SNF complex, which is involved in regulating gene activity by playing a role in remodeling how DNA is packaged in the cell. This process affects the repairing of damaged DNA, copying DNA, and controlling the growth, division, and maturation of cells. Through this process, just like the other genes we've discussed, the SMARCB1 gene plays a role in protecting against tumor cells. The LZTR1 gene provides the instructions for making a protein whose exact functions are unknown, but given its presence in several tumor types, the LZTR1 protein is thought to act the same way as the other proteins we've discussed, keeping the cells from dividing too quickly or uncontrollably. Just like we've seen before, mutations in these genes are inherited in an autosomal dominant manner, and one mutated copy is needed for a genetic diagnosis of schwannomatosis. Studies suggest that mutations in SMARCB1 or LZTR1 alone are not enough to trigger the development of schwannomas, and additional genetic mutations acquired throughout a person's lifetime may be required for tumors to form. The most common of these acquired mutations are mutations in the NF2 gene. Researchers also believe that other genes are associated with schwannomatosis, but are yet to be identified. A positive test indicating a disease-causing mutation in one of the genes we've discussed today, the NF1 gene for NF1, the NF2 gene for NF2, SPRED1 for Legius syndrome, and SMARCB1 and LZTR1 for schwannomatosis, will lead to the genetic diagnosis of the associated neurofibromatosis condition. Surprise! To make sure this information is clear, we'd like to ask you 2 questions to check your understanding so far. Please pause the video to see if you are able to answer the question before we reveal the answers. The first question is a case study. A 15-year-old patient presents with multiple café au lait spots, freckling in the armpits and groin, and learning disabilities. Their primary care physician is unable to find any tumors under the skin. Given these symptoms, their physician suspects NF1 and orders a genetic test. However, the NF1 test comes back negative. The physician reevaluates and orders a panel test for all the neurofibromatosis genes. Which gene is most likely to have a positive result, which means the presence of a disease-causing mutation? Is it A, SPRED1, meaning the patient has Legius syndrome? Is it B, NF2? Or is it C, LZTR1 or SMARCB1, meaning the patient has schwannomatosis? Please pause now. The correct answer is A. The patient most likely has a disease-causing mutation in the SPRED1 gene, meaning they have Legius syndrome. The next question is, the clinical feature most indicative of NF2 is A. Subcutaneous neurofibromas, which means tumors under the skin, B. Vestibular schwannomas, or tumors in the inner ear, or C. Chronic pain due to tumors in Schwann cells. Please pause now. The correct answer is B. Vestibular schwannomas, or tumors in the inner ear, are most indicative of NF2. Okay, welcome back. So let's say you receive a positive result from one of the neurofibromatosis genetic tests, meaning that you've been genetically diagnosed with one of the conditions we've discussed. Now what? As we mentioned before at the beginning, the main benefit of genetic testing is the ability to improve screening for tumors and inform family members so that they can be tested and diagnosed. Let's go through some of the screening and management options available, focusing once again on NF1. Screening measures primarily involve using MRIs to monitor the size, extent, and growth of neurofibromas, and to find and treat any brain tumors that occur, and monitor for the presence of optic gliomas, which are the eye tumors associated with NF1. Patients with neurofibromatosis conditions are also recommended to undergo periodic medical evaluations, including an annual physical exam, frequent ophthalmologic exams, regular blood work, developmental assessments for children affected with NF1, the MRI screening mentioned before, and cancer screening such as annual mammograms for female NF1 patients beginning at the age of 30. Please discuss these screening and evaluation measures with your clinical care team. Unfortunately, there's no cure for any neurofibromatosis condition. However, there are some measures for controlling symptoms. To complete our presentation, we will present some treatment options available to patients. Please note that these options should be discussed with your physician, as clinical management is often case-specific. Neurofibromas that are large or disfiguring, known as plexiform neurofibromas, can be surgically removed. Additionally, the drug Koselugo has been recently approved by the FDA to treat children older than 2 who have symptomatic clusters of neurofibromas that cannot be surgically removed. 66% of people who receive this drug show shrinkage of their tumors. Smaller neurofibromas can be removed by lasers. Most optic gliomas are asymptomatic and do not require treatment. If the gliomas progress, chemotherapy treatment might become offered to shrink them. Similarly, most brain tumors are asymptomatic and grow very slowly or not at all, and monitoring their growth is often sufficient and treatment not required. However, surgery may be offered in some cases, and chemotherapy may be offered to treat inaccessible tumors. Finally, any severe bone deformities in children with NF1 often require surgical management. That brings me to the end of our presentation. We hope you've learned a lot about NF1, NF2, and Schwannomatosis, and have a better understanding of why genetic testing for one or more of these conditions could be suggested for you. We encourage anyone referred to genetic testing to pursue it, as it can help guide your clinical management and inform family members. Thank you very much for your attention, and please see the next slide for additional resources.